Dr. Ingrid Corazzari graduated Pharmaceutical Chemistry and Technology at the University of Torino and is currently a postdoctoral researcher engaged in the research of asbestos and other toxic particles with a project aiming to assess the toxicity of celestial dust and irritants in response to future planetary exploration. She is a member of the ESA's topical team on celestial dust toxicity. Dr. Ingrid Corazzari, we're ready for your speech.
Thank you for inviting me to this Congress, and I want to present an overview on the history and the future perspectives for the evaluation of toxicity of lunar dust, which is an important problem for future exploration of Moon and also Mars. In the first direct experience of humans with celestial dust occurred in 1969 when the Apollo 11 crew landed on the Moon. This mission was followed by 5 more crew landings, and the last Apollo mission was Apollo 17 in 1972. Even though all those missions had different goals, they shared some common objectives that induce exploration and the examination of the lunar environment, the assessment of the feasibility of establishing a lunar outpost, and the collection of soil and rock samples. All those missions shared also another aspect that was the adverse effects of the fraction of the lunar soil with a diameter smaller than 20 micrometers, which is known as lunar dust. Lunar dust caused a variety of problems to equipment and instruments, including seal failures, contamination, and the abrasion of the spacesuits, especially gloves, during the extravehicular activity, the EVA. And moreover, lunar dust caused also a variety of health effects to astronauts upon inhalation and upon contact with nose-mucous membranes and eyes. So the information on the exposure and the health effects suffered by the astronauts are all anecdotal, and can be found in the debriefings that crew members released after each mission. And in particular, it's reported that lunar dust exhibited peculiar capability to adhere and to contaminate everything, every surface, and the capability to enter the inside of the spacesuit. The dust was easily airborne when the suits were removed and was inhaled or came in contact with the eyes, skin, and nose. After the exposure, astronauts suffered several effects, including symptoms of respiratory irritation and hay fever-like outcomes. These symptoms were named lunar hay fever. These effects were characterized by cough, sneezing, nasal congestion, and so on. There is also a curious feature of moon dust, that is its pungent smell, which is similar to that of burnt gunpowder. These features suggested the presence of some volatile species on the surface of dust particles. It is noteworthy that all these symptoms were transient and no lasting effects were observed in returning Apollo crew members. This is likely due to the brevity of the exposure. So the Apollo program ended in 1972, but crewed planetary exploration has remained of great interest for NASA and also for several other international space agencies, including the European Space Agency. With the Artemis program, NASA plans to bring a human crew on the moon by 2024. With the aim of establishing a permanent human presence on the moon and to create a sort of outpost for the future exploration of Mars. The Artemis program envisages long-duration missions, planetary operation, including EVA, and in situ resource utilization. That entail several risks. One of the major concerns for future crew missions is represented by the celestial dust. So in this figure, there is represented a quantitative estimation of the lunar dust exposure for each step of crewed activities during the Artemis program. This estimation was made by the topical team of celestial dust toxicity, and it is evident that during the early crewed activities of phase 1, the exposure is expected to be very limited because of the brevity of the missions and the low number of the astronauts involved. Also, the first steps, step 2 of phase 2, will be characterized by a limited presence of humans on the moon, but the entity of exposure is expected to increase with respect to phase 1 due to the suspension of a relevant amount of dust during landing and ascending activity. Dust exposure will significantly increase at the later stages of phase 2, when the residence time of astronauts will increase up to and potentially beyond 1 month. And the number of activities on the surface will be significantly higher than that of the Apollo missions. Moreover, the creation of sustainable infrastructure to explore and sustain human life on the moon will be achieved by in situ resource utilization strategies, which can represent a possible exposure scenario. Furthermore, the dustiness of the lunar habitat will significantly increase with the expansion of the exploration activities. In analogy with the moon, a similar picture can also be considered for the future exploration of Mars. So the exposure to celestial dust can occur during routine EVA, transfer from the lunar surface to habitat and vehicles, activities during a contingency situation, and the engineering failure of the dust system. This failure includes also the contamination of the inside of the spacesuit and or of the module or the habitat after extravehicular activity. So even though future habitat will almost certainly include some system, some airlock, which will reduce the entry of dust accumulated on the suits or on the equipment, it is very likely that any activity will bring with it dust exposure that will require some mitigation strategy. So the route, in the context of the exposure scenario, there will be variety of exposure routes, identifying the route through which celestial dust can enter the body in the context of the possible exposure scenarios, big of importance to predict which tissues, organs, and system could be damaged. If the expected effect will be local or will be systemic, all this consideration, all this information will be available in the planning of most efficient countermeasure to prevent any detrimental effect. The most important route of exposure is inhalation, because inhalation of airborne dust can occur very likely, especially during the removing of a contaminated spacesuit. Inhaled dust can reach the lung epithelium or can act on the nasopharyngeal or oropharyngeal mucous membrane. The dust could enter into the body also through other routes, including skin, eyes, and by ingestion. The exposure through skin can occur via traumatic injury or a penetrating injury resulting in a wound that becomes contaminated with dust. Dust can also enter through minor wounds or abrasion when spacesuit abrade the skin. Moreover, if celestial dust enter the suit interior, as was the case of the Apollo missions, this could induce abrasions or enhance the suit-induced injury. There could be also ocular exposure that can occur during the removing of the contaminated spacesuit, and the ingestion of dust, which can contaminate the food during storage or preparation. And we must also consider the possibility of ingesting dust by eating edible plants grown using planetary soils. Eating vegetables grown with terrestrial soils may be a way of direct ingestion of celestial dust, but also ingestion of toxic ions leached by irrigation water and absorbed and concentrated in the edible plant. At the light of this, the need to study the toxicity of celestial dust becomes of paramount importance. What we know from the literature on earth mineral powders is that there is a variety of physico-chemical properties that can make a dust toxic. The picture is very, very complex, but can be summarized as represented here. We have bulk properties. Among bulk properties, we must consider shape of particles. For example, fibrous habit is associated to the toxicity of asbestos. The crystalline structure, crystallinity plays a role in the toxicity of quartz. The size, nanoparticles can be more toxic than the micrometric counterparts. And on the size depends the capability of an inert particle to reach lungs. Another feature is the surface reactivity. Regarding surface reactivity, the interaction of a particle with the biological matter depends strictly on it. And the surface reactivity can be influenced by the surface chemistry, for example, the presence of reactive centers. And surface reactivity can be modified and altered by the biological surrounding medium. And this alteration can result in a decrease in the reactivity and toxicity, or at the contrary, in an enhancement of them. Finally, an important aspect is the biopersistence, because biopersistence plays a key role in toxicity. It indicates how long a particle remains in contact with the cells and tissues before being removed by clearance mechanisms or dissolved. The longer a particle remains in a critical biological compartment, causing stress, oxidative stress, and damage to cells and tissues, the greater the adverse effect expected. So one most widely recognized mechanism of toxic outcomes of mineral particles is the oxidative damage, which is described here in this figure. There is a first path of the oxidative damage, which is the direct oxidative damage in this particle. This is mediated by reactive sites at the surface, which can trigger the generation of reactive oxygen species, ROS, derived directly from the particle. These species include hydroxy radicals, superoxide ions, and singlet oxygen. The oxidative damage can be also induced by reactive sites that cause them by direct interaction with biomolecules, such as membrane fatty acids. And another mechanism is the release of redox active metals, which can be involved in the release of reactive oxygen species. And this overproduction of reactive oxygen species can induce the depletion of endogenous antioxidants, and also this can be a cause of impairment of the cell homeostasis. There is a second path, which is mediated by the ROS derived by cells. In this case, the damage is induced by the overproduction of reactive oxygen species from the cell after the activation of the immune system or the alteration of the mitochondrial function. This figure represents the reaction at the molecular level. So we can have a reaction of production of hydroxy radical, which is a very reactive species via Fenton-like reactivity, or we can have the direct interaction of biomolecules with reactive centers. We must consider also health effects that can be mediated by other mechanisms. As an example, the release of ions can cause sensitization, which exert allergic reactions, or ions can interact specifically with the receptor or ion channels, inducing the perturbation of specific functions of tissues and organs. We must consider several characteristics of celestial dust, which can be determinants for toxicity. In this slide is reported the elemental composition of several Apollo samples, which were studied thoroughly and characterized in terms of chemical composition, mineralogy, size distribution, and shape. There are some peculiar features of real lunar dust, which can be relevant to toxicity. The first one is the presence of zero-valent iron, which is a peculiarity because this feature is not present on terrestrial dust. In particular, the fraction smaller than 5 micrometers is rich in this nanophase iron, which is formed during vapor deposition caused by the impact of micrometeoroids. The abundance of this nanophase iron increases with a decrease in particle size, and its presence is relatable to the high reactivity of lunar dust and may play a crucial role in lunar dust toxicity. Another peculiarity is that the remaining iron in the lunar dust is iron 2. There is no iron at high oxidation state. And also this feature could impart a peculiar reactivity which can exert oxidative damage. Moreover, in lunar dust there is ilmenite, which is a mineral that is present also on Earth, but ilmenite on the Moon is quite different because it doesn't contain iron 3, sorry. Is different from the ilmenite on Earth. And this difference is relatable to oxidative, a higher oxidative reactivity in lunar dust. Moreover, ilmenite is also associated to the release of oxygen-free radicals upon irradiation because he possesses some photocatalytic properties. Furthermore, there are several environmental conditions present on the Moon that can enhance Moon dust toxicity. There is a continuous micrometeorite impact, which causes the continuous formation of freshly fractal surfaces that could be characterized by high reactivity. The surface activity is preserved because of the absence of an oxidant atmosphere. The solar wind and the space radiation continuously modify the surface of the particle, inducing electronic defects and the implantation of ions. Also, this can result in the high reactivity centers of the surface of the particles. Moreover, the condition of reduced gravity thus becomes very easily airborne, and this increases the probability of contamination and of inhalation of these particles. So concerning Mars dust, the investigation of Mars dust nature was carried out in situ during robotic exploration. This table report the elemental composition of Mars soils. Also, Mars have a chemical feature that can be relevant to toxicity. There is the presence identified or suggested of oxidant, including iron-bearing species, reactive oxygen species that can be involved in toxic outcomes, but the most evident peculiarity is the presence of high amount of perchlorate, which is toxic ion. Perchlorate seems to be ubiquitously distributed on Mars surface. It's extremely soluble in water, can be absorbed, concentrated by plants, because it's not toxic to plants, to vegetable, but it's toxic, extremely toxic to humans if ingested. Its mechanism of toxicity is the competition with iodide for the iodide pump of thyroid. So there is a decreased uptake of iodide, which induces the decrease of thyroid hormones output. For the earlier stages of the exploration, it's not so likely that perchlorate will be heated, will be ingested. But when the permanence of astronauts will be established, it is quite likely that to support the habitability, edible plant will be grown in situ, employing and employed by astronauts as food, with risk of the ingestion of high doses of perchlorate. Some mitigation strategies are proposed to resolve, to remediate the pollution of perchlorate Earth, because perchlorate is also present as pollutant on Earth, and they possibly could be also applied, some of them, in the future to eliminate perchlorate from Martian soils. Celestial dust would be the ideal material to carry out studies in a toxicological perspective. On the other hand, there is only limited amount of real lunar dust brought back on Earth during the Apollo missions to be widely distributed to the scientific community. And the real samples of Mars dust are not yet available, naturally. For this reason, there is the necessity to use simulants that can be available to the wider scientific community to carry out both reactivity and toxicological studies on this dust. And for this reason, there is the necessity of create celestial dust simulants that mimic the real celestial dust with a high fidelity, and that can be distributed wider to the wider scientific community. Here's some example of simulant. There is a variety of celestial dust simulant that are widely employed. These are simulants from NASA's, which are of natural origin, and similar from Exolith lab that are prepared by starting from raw mineral components. A few of the dust which are declared to be simulants are recorded as dust simulants, really. And because the hazardous and the toxic properties that are typically reduced for the purpose of safer human handling. And so it is clear the need for developing new dust simulants, especially for long-term toxicity studies. So how to obtain an high fidelity simulant for studies in a toxicological perspective? The idea is to start from a root simulant. It must be standardized, and it must share with the real dust the chemical composition, crystallinity, size, surface area. And this root simulant will be modified depending on the purposes of the research. Grinding, sieving is required to gain the proper size. And in the case of lunar simular, to mimic also the continuous action of micrometeorites. A key point would be the addition of specific components that are present in the real dusts. As an example, nanophase iron for lunar dust simulant, or perchlorate for Mars simulant. Moreover, the irradiation with high energy radiation is recommended because of such condition, which occur especially on the moon, is an important key factor that can influence the reactivity of the dust. So the current knowledge on the pulmonary toxicity derives from several experiments. I reported here some of the most important findings. That pulmonary toxicity in rats demonstrated that real lunar dust toxicity is an intermediate between titanium dioxide, which is employed as a negative control in quartz, which is characterized by a high pulmonary toxicity. On the basis of in vivo experiment, NASA set 0.3 milligram per cubic meter as a preliminary exposure limit during the future mission during the Artemis program. So there is also evidence from the literature on the dermal irritation, but the literature is very limited, and evidence is that both real dust samples and a simulant, the JSC-1A simulant from NASA, are able to induce abrasion of a pig skin, which is chosen as a model, similarly to what observed in sandpaper. There is also limited literature on eye effect of lunar dust, but the literature did not evidence very severe effect. The irritation observed with in vivo, employing both in vitro model and in vivo model. In vivo model was observed only mild and transient irritation. And for this reason, there is no special precautions that were recommended. Okay, but there are several gaps in the knowledge of CD toxicity. The most important major gap that must be fulfilled in view of the Artemis missions is the effects of the long-term exposure. Because, of course, during the Artemis missions, the presence, the permanence will be long-lasting. So the knowledge, the current knowledge on the oxidative reactivity, evidence that the oxidative reactivity of the real lunar dust in comparison with the JSC-1A simulant, evidence that the real lunar dust have a higher oxidative reactivity than simulant, and this reactivity was attributed to the release of hydroxyl radical by Fenton reaction. And moreover, the presence of nanophase iron was related to the high reactivity of real lunar samples. Okay, this is referred to the reactivity of lunar dust, which is relatable to the presence of iron and to the release of hydroxyl radical. We try to clarify the mechanism of the oxidative reactivity of celestial dust. And to do it, we chose the terephthalate assay as a probe molecule. Terephthalate assay is a non-fluorescent molecule that when it is oxidized to hydroxyterephthalate, for example, by hydroxyl radical, becomes fluorescent and can be measured by means of a fluorimeter. So this molecule is usually employed as a dosimeter to measure the production of hydroxyl radical. We employed this assay to measure the oxidative reactivity of JSC-1A, very fine simulant. The results show that JSC-1A was able to oxidize terephthalate. The experiment was also carried out in the absence of oxygen because we want to clarify if the observed oxidation was involved oxygen derived free radicals derived from the interaction of JSC-1A with oxygen dissolved in the solution. We will find out that JSC1A was able to oxidize TA also in the absence of oxygen. And this indicates a mechanism that does not involve oxygen-derived free radicals. Try to identify the species, the radical species, which was involved in the observed TA oxidation. We employ EPR spin trapping using a Fenton system as the positive control and buffered suspension of JSC1A very fine in the presence of a spin trap molecule. This technique, which allow to detect paramagnetic species, did not evidence any radical species released in the suspension of JSC1A-UF. So this result confirmed that no oxygen-free radicals were involved in the reactivity observed toward the terephthalate. Oxidation of our probe was mediated by something else than free radicals. We hypothesized that the oxidation of T8 was possibly mediated by reactive centers at the surface of the dust particles because this is a characteristic that is common with other mineral dust. In particular, purely coordinated iron 2 at the particle surface can impart these oxidative properties. And to investigate the possible role, we employed ascorbic acid, which is able, following this mechanism, to reduce iron 3 at the surface to iron 2. We observed adding the ascorbic acid to a suspension, a buffered suspension, of JSC1A, the presence of ascorbic acid, which is, and the reduction of the iron center induced by ascorbic acid. In this condition, the amount of TA oxidized was higher than in the absence of ascorbic acid. So this confirmed our hypothesis that this lunar simulant oxidize TA through mechanism which is driven by iron 2 at the surface. Okay, the hypothesis on the basis of some studies of asbestos reactivity, we hypothesized that the higher oxidative reactivity of observed was mediated by the formation of iron phosphate complexes, because in our suspension, in each experiment that we carry out, the dust is suspended in a buffered, in phosphate buffered suspension of terephthalate. So we hypothesized that the high oxidative reactivity was mediated by the formation of these complexes, which can act in our experimental condition as oxidizes pieces following some redox cycle where also ferryl ion is possibly involved, but this mechanism is not clarified. Test the capability of JSC1A to in the oxidative degradation of membrane lipids, which is a recognized cause of damage to cell membrane, which occur following a chain reaction that can be triggered by free radicals or by interaction with reactive center at the surface. We employed the linoleic acid as a molecule of membrane fatty acid, and we measured the degradation by means of TBA assay. We observed that JSC1A was able to induce the degradation of linoleic acid, but that the addition of ascorbic acid induced a higher reactivity, and a higher amount of linoleic acid was degraded. And this indicates that also the reactivity, the reactive species, involved in this activity are likely iron 2, analogously to what observed with the terephthalate. Capability of this test to measure the overall oxidative reactivity of mineral powders, which can be significant for the toxic outcome, and the easiness of execution make this test suitable for in situ experiment during planetary exploration. One of the major problems in the employment of TASA during planetary exploration, in situ exploration, is that this test requires an aqueous environment to work. We know that the employment of water can be a big problem, the environmental conditions of Moon and Mars, because we have a lower atmosphere, lower gravity than on Earth, and this can influence the dynamic of fluids. And so to overcome this problem, it was conceptualized a particular system which is called a TA patch. This was an idea of some of us topical team on celestial dust toxicity. This patch contains the terephthalate dispersed in a buffer hydrogel. So this condition ensures the aqueous ambient environment without the employment of liquid water. This system is sealed. After removing the seal, the TA is contacted with the dust sample to allow the reaction with TA, and the hydroxyl terephthalate possibly produced can be measured by a simple fluorescence microscope. So this approach could be completely automated and inserted in a multi-instrument suite, like that's sketched in the slide, and employed during the activities of planetary exploration to measure the oxidative potential of celestial dust in their environment in situ. In conclusion, the prediction of celestial dust is paramount importance to protect the astronauts and plan efficient mitigation procedures. And for this reason, studies are required to predict toxicity in order to mitigate the effect of this dust. There are many gaps in the knowledge of CD toxicity that must be fulfilled in view of human planetary exploration, and the major gap is represented by the effect of long-term exposure. Since real dust are very precious, not yet available, simulants with high fidelity are required to demonstrate the robustness of techniques and experiment approach. And the toxicity assessment using authentic dust may be possible only after the successful demonstration of the feasibility and the robustness of the technique. The employment of easy-to-perform chemical tests could be an efficient tool to perform experiments in situ to clarify the real CD oxidative potential. The research group at the University of Torino and is a T3CD. And thank you for your kind attention.